Hello? Eduardo? Tá ótimo. Eduardo, can you see the screen? I think Eduardo's sound is not on. O som dele...
Vê se ele põe aqui, no verde. Participantes. Tá normal, tá faltando... Vê se... Ele não falou nada, falou?
No, I think he's saying something, but...
O som tá... Ah, tá. Vê aqui, ó. Isso aí. Aperta aí. Aperta aí de novo.
Projetor?
Vai, pede pra ele falar alguma coisa.
Eduardo, can you hear us?
Mudou. Pede de novo.
Can you say something now? We changed the... No.
Não? Assim, então. Vamos lá.
Let's try one more time.
Creston.
Oops.
No problem.
Okay.
Isso aí. É o Creston, isso aí. Pede pra ele falar agora.
Okay, let's try again. Eduardo, can you say something?
Yes.
Okay,
Can you
now
hear
it's
me?
perfect. Yeah, perfect. Great. Okay. So you can see the screen, right?
Yes, I can.
Okay, perfect. Thank you. Okay, Nathan, we can
It's
start
pretty
now.
good.
Okay. Hi, everybody. Thanks for coming again. Yesterday, I've told you about big projects that were basically complete. Today, I'd like to give you some more ongoing an idea of some more ongoing stories that are closer to experiments. And that, I think, will give you an idea of how I think of interaction between biological facts and what theoretical physics can bring to that. And I'd like to tell you a story about a class of bodies that are found inside cells, inside living cells, that has been identified pretty recently, and that is sometimes called membraneless organelles. So if I make a cartoon of what a cell is with its nucleus, it has a number of intracellular bodies, sometimes called organelles. For instance, the mitochondrion, which is somehow the powerhouse of the cell, the Golgi apparatus that does sorting of proteins. There's also, of course, the nucleus, which contains all the genetic information and all the chromatin and does a lot more. And there's been another class. So all of these organelles are separated by membranes. They're compartments that have an inside and an outside, and that ensures their stability and the fact and allows to have a different chemical environment inside and outside. Now, in the past 11 years or so, there's been a lot of interest about a new class of intracellular bodies that comes as apparently spherical objects in the nucleus or inside the cytoplasm, and which have the specificity of being membraneless. So they're like a phase-separated liquid droplet they're not compartmented away from the rest of the environment. There's a multitude of them. There's, I think, about 20 different classes of them that have been identified since. Some of them are extremely important, such as the nucleolus that is found inside the cell nucleus, and where ribosomes are assembled. So ribosomes are the machines that convert RNA, genetic information, into proteins, and that they're assembled in these membraneless organelles. Other ones, such as stress granules, that appear perhaps as a kind of storage when the cell undergoes chemical or thermal stress. And another one that I'm going to tell you a bit about is called the pyrenoid. It's a huge organelle that is about 10 microns inside. And it's found inside unicellular algae, so plankton, and it helps doing carbon fixation. And this one too is liquid, and it helps to do about half of the oxygen production on Earth. So this is a like expanding list. There's a number of known facts about this class of organelles. They nucleate and dissolve. So nucleate meaning that they appear as a point that grows when they form. They can merge, they can fuse and coalesce. Meaning that if you have two of them that come close together, they're going to merge. And then in the long run, they will make one larger round droplet. If you shear the cell, they're going to deform on this door. If you look at the diffusion of one particle inside these droplets, it diffuses freely. If you look at the molecular structure, there's no crystalline order. It's pretty disordered, much like a molecular liquid. And so all of these observations tend to point towards the fact that these are liquids. And there's sometimes in the introduction of papers compared to oil in water, in that their internal interactions that allows them to phase separate from the rest of the nuclear of the cellular environment. And so
oil and water is a tough comparison.
Yes.
And the reason for that is we see that in water phenomena, because if you phase separate not small molecules, minor variations create a phase separation. Oil and water, you have an enormous amount of loss of configuration entropy when they say separate, but they're polymers, small amounts. So you get large polymers, they're just slightly different, they start to phase separate. That's why these problems are even more complicated than oil and water.
Thanks a lot. So I'm going to announce the plan of this talk. The first part will be not like oil. And the second part will be not in water. And I hope this will resonate with what you just said.
Let's see.
So if you start with this observation, it's a fair question to ask what a theoretical physicist might bring to this picture. The physics of liquid-liquid phase separation is well characterized. It's been known for more than 100 years. And for a while, I've turned around this field and observed the experimental talk. I thought, OK, is there anything I can do here? And that was during my whole time in Princeton. This is a very hot topic in Princeton because the person who discovered these phase-separated bodies is Cliff Brangwyn, who is a professor in Princeton. So now there's about 5 or 6 experimental groups dedicated to studying these at Princeton. And in the past few years, I think I've identified a few questions where indeed a theoretical physicist could add something to the picture and make prediction and perhaps even surprising predictions about what these organelles might do. So the first part I'd like to tell you about is why the internal interactions inside these droplets are very different from usual liquids, and how cells might use these specificities to tune the properties of these droplets. The second part will have to do about the interactions of these droplets with their physical environment, which has nothing to do with water. It's crowded. There's a DNA, there's the cytoskeleton, and so you have to take into account of the mechanical and chemical interactions that there are at this point. So let me start with the start and take a look at first what is a usual liquid. Okay, a usual liquid is made of many particles that interact with short-range interactions. And essentially, these short-range interactions determine most of what you can understand of this liquid. Its cohesion, the distance between particles, they're going to be close packed so that they optimize this molecular interaction. And each molecule is going to try to get as many partners as it can in order to minimize its local energy. Now, these are for instance, these are the typical interactions for simple liquids. In protein liquids, there's at least 2 different paradigms for protein-protein interactions. The first one is called intrinsically disordered proteins. And these are proteins that do not fold into a specific shape, but retain their polymer, their random polymer chain physics. And these are able to make many weak interactions with each other, and they're thought to be of crucial importance to a large fraction of these phase-separating droplets. And I don't have too much to say about those. I'd like to focus about a second paradigm for protein-liquid phase separation, which is specific interactions between complementary protein domains. So these ones are relatively weak, numerous interactions that have somewhat low specificity. Here, I'm going to be talking about...
So you're going to only use protein. You're going to put RNA on the game.
Yes, there's also RNA in the game. The systems of which I'm going to talk about which is essentially this one, the pyanoid of algal cells. I don't think DNA is a key RNA is a key component of that droplet.
There's RNA on this, more
What?
super enhancers and lots of other things.
Yes, I'm going.
Probably better if you summarize the question, otherwise there's a lot of
Oh, yes, sure. So there are many different components inside these droplets. Some of them there's been up to 100 different chemical species found inside the droplet. There's RNA, there's a lot of different types of proteins. It's unclear at this stage which ones are truly part of the scaffold that phase separates and which ones are kind of clients that get recruited into the organelle but do not contribute to the phase separation. I'm going to focus on a class of simplified systems in which there's really two species that control the phase separation. For instance, and that do that with complementary protein-protein interactions, a bit like the lock and key interactions that folded proteins generally have. And I'm going to represent this as short polymers, which have a number of complementary binding sites that I'm going to represent with different colors, and such as these ones. And here the yellow cup domains will want to bind strongly with the red domains. So these are well-identified domains that are complementary, and that provides many kT thermal energies of binding energy for each bond, typically 5 to a bit more. But they're specific. So once you're bound, and that interaction saturates. So in contrast, this one will want to make a gel in which you have as many bonds as possible that I'm going to represent like this. And so in contrast with, each of these bonds bring a lot of energy, so you're going to try to satisfy as many of these as possible. And there's a strong contrast with usual liquids, which is that once a bond is satisfied, there's still a lot of remaining freedom. Well, here, you needed to get as many neighbors as possible, and that leaves very little remaining freedom. And so this has a number of interesting consequences for the physical properties of these liquids. The first one I want to mention is the importance of these specific interactions on the phase boundary of these liquids. So if you ask what is the phase boundary, so whether these proteins will phase separate and form a liquid droplet, or whether they will remain soluble in solution, you have to compare two scenarios. That's the solute case, and that is the dense case. So here you have a number of single molecules. And here you have basically all the bonds that are satisfied within the gel. And so in order to compare these two scenarios, you need to realize that here you have very high energy. You haven't made any bonds. But there's very high entropy because each of these proteins is able to diffuse around, and so there's a lot of translational entropy for these. If you want, you don't know anything about their position. Here, they're all colocalized. They're all within the same droplets, so there's low entropy. And there's very low energy because you've satisfied all these
You're calling
bonds.
entropy here just for completeness. That's configuration entropy, right? You're not talking about solvation, dissolution of these polymers, just so that we have the numbers correct.
I'm not going to put many numbers on these.
Right,
but
but there are two numbers that comes on the entropy, right? One is this configuration entropy they want to use on folding all the time. But also when these polymers come together, they're going to get dissolvated. Yes. So depending on what your binding interactions are, that these numbers can do a big contribution, different signs. But
So one comment I can make about that is that even in the condensed phase here, you have a pretty low protein content. It's up to 10, 20%. So there's still a lot of water. The polymers are still solvated.
Okay, but okay.
So you have low energy and low entropy here. And so you want to compare the two. And because I've said that the binding energy is very high, so you gain a lot of energy when you bind, you would tend to think that this is the favored phase. But actually, there's a different dilute phase that is possible, which would consist in, so this phase is disfavored because of these strong binding interactions. But you can think of another dilute phase in which you would make small dimers or trimers or multimers that satisfy all the possible interactions. And so gain a very, so have a low energy. But now a slightly lower entropy too, because you've colocalized these bonds. And so comparing these two phases, oligomer phase in which you have small complexes, which are not necessarily folded. They can be randomly arranged, or this high density, completely disordered liquid phase. Now it's more complicated because the energy is the same. They're all satisfying all the bonds, and you can only compare the entropies. The way these entropies are going to compare depends actually on a crucial quantity, which is the relation between the valence of the red and the yellow polymers. If the red and the yellow polymers have the same length, you can form dimers, which are pretty favored, and retain a lot of translational entropy while already satisfying all the bonds. So if you look at a phase diagram in the concentration, temperature, or binding energy, inverse binding energy phase, if you have, for instance, I'm going to assume that there's 4 binding sites on the red. If you have 4 to 4, you're going to be very stable, and the phase boundary is going the phase separation is going to occur at very high concentration, even when the temperature is very low, because you can still, at 0 temperature, you can still satisfy all the bonds by forming these small oligomers. While if you have a mismatch, so if you have one more site on the red than the yellow, or one less, then you have a dangling end that will want to stick to something else from larger complexes. And because the numbers are not commensurate anymore, you will lose a lot more entropy if you want to satisfy all the bonds. And so what we've observed with molecular dynamics simulations is that if you look at the 4-3 or the 4-5 curves, phase boundaries, the phase separation is much more favored than in the 4-4 case, in the equal valence case. So you have a rather striking effect here, in which the effective strength of the interaction, which is controlled by the number of bonds, does not control in a monotonic way the phase boundary. Changing the valence can lead to jumps in the phase boundary depending on the size of these polymers. And we suggest that this is an effect that these algae, Chlamydomonas reinhardtii, can use during mitosis. During mitosis, this pyrenoid dissolves, and so it dissolves and fills the whole cytoplasm. And then after mitosis, it renucleates in both daughter cells. So what we suggest is that because the molecular structure of the proteins involved seems to match with this magic number effects of
So you buy fully the magic numbers from Ed.
Say again?
So you believe on Ed Wingreen magic numbers?
I think it's yes. I mean, I co-signed that paper, so I somewhat believe in it. Yes.
Can you summarize what the question was?
So he asked whether I buy Ned Wingreen's magic number theory. This is Ned Wingreen's and myself and others' magic number theory. So this is at least a way in which cells can use these strong. Okay, if you look inside this, this organelle, it's made of large proteins, about 10 nanometers, which are called Rubisco, which incidentally are the most abundant enzyme on Earth, and which have 8 identified binding sites, which bind to another linker protein that has 4 to 5 of these of complementary binding sites with these. And one of these 5 is different from the 4 others. And there's a theory that has not been experimentally confirmed yet, that one of these might get phosphorylated, and so activated or deactivated. If it's activated, you have 5 to 8, so these numbers are not commensurate, and so you will be in the phase-separated case. If you suppress one site, then you can make trimers, which are fully bound and still diffuse around.
So the last time I heard about that, they were doing the phosphorylation experiments. Have they been done or not? They're commenting about it. I don't know if it was.
Yes.
I heard net about a year ago or something like that. So I don't know if there's.
Yeah, I don't think there's been major progress on this in the past year.
Well, it's a pretty cool idea if it works.
Yeah,
right. It would be nice to see.
It might work or not.
Okay.
I wouldn't say that I'm a strong believer. It's a nice theoretical tool. It's a nice tool that cells might use here or elsewhere in order to control phase boundaries. I'll stick to that.
Sorry, you get interrupted from the story. So can you just go from the story from the middle to the end so I can see?
Okay, so the story is depending on the ratio between the valence of the 2 complementary proteins, you are going to phase separate to favor phase separation if the numbers are incommensurate, or disfavor phase separation and favor a phase of multimers that's
right, but
free
like
diffuse.
you said. Oh, I'm sorry.
You ask more advanced questions. You never get back to the basics.
There's also other things that I want to talk about that are more recent. So I'm going to wrap up on this part of the story. Changing the valence of these of one of the 2 proteins can shift dramatically the phase boundary. And if you're here, for instance, on your 4-5, you're going to be phase separated. And if you switch off one of the linkers, suddenly you're not phase separated anymore, and you're going to dissolve completely.
The thing is that the cell somehow does this shift.
The idea is that the proposition that's really at the speculation level is that just before phase separation, this pyanoid dissolves at 90%. There's still a small punk that remains, and then it phase separates. It divides into 2 daughter cells, which re-nucleate the pyanoid. The idea is that they might use this mechanism, which is compatible with the molecular structure, or not incompatible with the molecular structure of the proteins that play a role here. That was the magic numbers. That's a proposition of how cells could use these strong specific interactions. I'd like to move on to another way these proteins can use strong specific interactions.
Maybe let me just ask a question. So, is it none of this is in the thermodynamic limit, right? It's crucial that it's relatively small numbers of
No,
polymers involved?
It is in the thermodynamic limit. There's many of these, but that are relatively small. So you're in the thermodynamic limit in the sense that there's many proteins, but they're not of large size. They are finite size.
But then, a priori, why is it crucial that they are commensurate or not? I mean, I can put several of 8 and several of 5, and eventually they would match, and they would still form.
Yes, but the numbers will be different. The entropic, you pay, binding two proteins together, you pay a translation cost for one of them. Suddenly, you've killed 1 degree of freedom, which is the translation of one of them.
Yes.
Binding 40 of them, like if you have 5 and 8, you need 40,
No.
So you need 40 binding sites. So you need 13 protein togethers. So you need to pay 12 times the translation cost. That's very different from 3 times. So
but is it because, that's why I was asking about the thermodynamic limit. Is it because those numbers are starting to be comparable to the total number?
No, it's not. It's because
if it was 1 million proteins, they would still not like to do it?
This dimer phase is high. The stability of this multimer phase is highly dependent on the precise valence that they have, in a way that does absolutely not disappear in the thermodynamic limit.
Thank you.
Moving into so that was for the phase boundary. Now I'd like to tell you a bit about what's happening in the bulk of these protein condensates. And this was thermodynamics. I want to tell you a little bit about dynamics. How do these proteins diffuse inside a protein droplet? How do they relax? And in a way that is connected by thermodynamics, what is the viscosity of these droplets? So the elementary move to get such a liquid to relax is to break bonds. You need, because you have strong specific interactions, you need to break a bond in order to get relaxation. Now, that could be the end of the story. So if I'm calling A the yellow ones and B the red ones, the elementary move would be a bond, AB, gives A plus B. Okay, I think I'm going to stick to white for this.
It's fine. You can't see the red.
Okay, so that's the elementary bond. Once you've broken a bond, well, these two new binding sites are going to diffuse around. But what's different from a typical liquid is that once they're unbound, they're still attached to other monomers which are bound, because this A and this B are part of polymers that have other binding sites, and so they cannot freely diffuse around. So they have a small volume that they can explore, and if they can't find a new partner in this small volume, they're going to bind again and cancel the move. So you have to consider 3 different scenarios. Either A and B get back together, or B gets a new partner, or A gets a new partner. And only these 2 events are actually a relaxation event. If you do this, you've gotten back with your X, and nothing has changed, and there's no relaxation. So what you need to evaluate in order to quantify the relaxation time of these liquids is the rate at which this whole step takes place. And so if there's a strong excess of A or a strong excess of B, this part is trivial, because there's so many fish in the pond. Each time you unbind, you're going to get with a new partner. But if there are very little fish in the ponds, then so if there's almost equal stoichiometry such that there's no excess of A or no excess of B, then the rate of this elementary step is going to be high. So if I call epsilon the binding energy, of an AB pair, what I find is that if you look at the viscosity, or the 1 over the diffusion coefficient, as a function of the stoichiometry, concentration of A minus total concentration of B, what I predict is that there's a huge bump exactly at around equal stoichiometry, which has height exponential epsilon over 2, and width exponential minus epsilon over 2. And so because epsilon is several kTs, this is a relatively large number. It can be tens to hundreds. And so there's a very sharp peak of viscosity close to equal stoichiometry. That's just a consequence of having complementary interaction between these proteins. I've made molecular dynamic simulations that confirm the scaling. And I think it's pretty ripe for experimental verification. I think the biological relevance of this is that it provides an additional knob that is not criticality. It's a simpler mechanism that allows a cell to, by changing sensitively the composition of its organelles, it allows to dramatically tune the mechanical properties, their mechanical properties. So this is, I think, a relatively nice prediction. That's a result I'm working on now.
Can you just remind of the connection between the effectiveness of this process
Yes.
and the viscosity?
So,
With coupling, the viscosity is very big, right?
so when you're at strong binding energy, it takes a lot of time to unbind thermally, and so the liquid will take a lot of time to flow. So the viscosity will be very high. And even higher if, on top of that, you're almost at equal stoichiometry, because in that case, the constant moves will be very probable, and so you're going to be around that peak.
Just out of curiosity, have you considered the possibility of some glass dynamics, these things bound and having to...
So one interesting consequence of this is that if you mix now two droplets, you have a viscosity that depends strongly on the composition. If I'm plotting it differently, if I plot the diffusion coefficient of this protein, as a function of the composition, then you'll have a sharp dip. And so you'll get this very strong composition dependence of the diffusion properties will lead to, I predict, aging properties in these droplets. Because if you start with pure A on one side and pure B on the other, then in both of these regions, you're very active. But at the interface between them, you're at equal stoichiometry, and you kind of freeze. So you're going to have strong dynamical heterogeneity of these, in these liquids. This is purely prediction.
So what you're telling me is that if you have equal stoichiometry, you're going to have glass behavior and aging. But if you go to a very separate stoichiometry, the system has enough freedom that is going to thermalize fast. That's what you expect.
If you're close to this regime, you're going to have strong dependence of the diffusion coefficient in the composition. And so thermal fluctuations in composition will lead to huge differences in dynamical timescales and some kind of dynamical heterogeneity. While if you're away from it, you're not going to have it. Yeah, that's a prediction.
So you thermalize if you're away from it. You think the time scales are long enough that you can believe you reach equilibrium. You're avoiding the words equilibrium and glass. I'm just trying to see if you're...
Sorry, can you repeat?
You're avoiding the words glass and equilibrium. I'm trying to figure out why this careful choice of words.
Because these are dangerous words. I'm going to say that relaxation towards equilibrium is going to be slow, and I'm not going to pronounce the word glass.
So something very protein-like, I mean, is this just some general chemistry, or is there something critical proteins are doing here?
So the question is whether this is specific to proteins or whether there's something more general only based on the chemistry. And the answer is, of course, this is only based on this assumption here. So if you make, actually one of the best testing grounds for this kind of predictions would be self-assembled DNA particles, which have very controlled complementary interactions and are much better characterized than proteins. The ingredient really is strong complementary interactions, strong complementary interactions between two different types of particles, and some flexibility that allows you to make use as much as possible of these interactions, so not too much frustration. One last thing I'd like to mention about this first part is the fact that I've talked about the bulk properties so far. There's another interesting question, which is what controls the surface tension of these droplets. Usually, in usual liquids, what controls the surface tension is the same pair interactions that control the bulk stability. It's just the fact that at the boundary, you have missing partners. And the cost of having these missing partners creates a surface tension and makes the droplets want to be spherical. Here, because of the flexibility of the proteins, you have no need to have missing partners at the boundary. You can still satisfy all the bonds and not have any unhappy bonds, or very few of them. So the surface tension is not controlled by the binding energy of this, the binding energy. It is controlled by the remaining entropy, so how much entropy it costs, or how much you have to stretch the polymers at the boundary to still satisfy all the bonds. And that allows for large tunability of the surface properties of these droplets too. And that's actually something that is observed, which is that these droplets have incredibly low surface tension, hundreds, thousands, tens of thousands of times lower than oil in water. So that was the first part about the properties of these droplets the internal properties of these droplets. Now I'd like to switch gears and tell you about the way they interact with the environment. And for that, I'm going to focus on a specific class of protein droplets, which are the nuclear ones, the ones that live inside the cell nucleus. So I've told you a little bit about the nucleoli that assemble the ribosomes. There's several other ones, one of which is speculated to be involved in RNA transcription. So whenever a gene is activated and gets into starts translating the DNA into RNA, you have transient small droplets that appear and that seem to have phase-separated the properties. Another type of phase-separated droplets that I'm going to use here is optogenetically controlled ones, so artificial droplets that are induced by genetic modification of these cells, such that when you turn on light, they start phase separating because it changes the formation of the proteins, which changes the binding energy, which make them phase separate. And these ones are a great way to probe the physics of this phase separation, because you can just turn on the light, off again, on again, and see what happens. You can draw any pattern inside a live cell nucleus, such as a smiling face or whatever you want. So the second part is not in water. And here I want to look into the interactions of these droplets with the surrounding chromatin, so with the surrounding medium. And here I've drawn a mesh like this. This is meant to represent chromatin, which is a whole DNA unpacked, filling pretty densely the nucleus. And the volume fraction of DNA inside the nucleus, so the volume fraction of this chromatin, is about 15 to 20% overall. So there's a huge density of polymers, relatively homogeneous with some important heterogeneities at some point. But there are no holes. So the question is, how does this affect phase separation? And how do these new droplets nucleate inside the nucleus? And for that, I'm going to start by looking at what happens in water. So how does a new droplet nucleate in water? So for that, there's a venerable framework, which is called classical nucleation theory. Which aims at assessing how hard it is to create a new droplet. And so if you look at a droplet of size R, you can ask how much free energy it costs or how much free energy you gain by creating a droplet of size R compared to the purely dilute phase. And the way this is generally written is that this free energy goes as a volume term, where delta G is the free energy per unit volume that you gain from phase separation because of the interaction, plus a surface term. So delta G is going to be negative in order to have phase separation because you need to gain free energy, plus a surface term for pi gamma R squared. And that's the surface cost, that's the surface tension associated with creating an interface at the boundary of the droplet. And so there are two scenarios. If you look at f of R as a function of R, the first scenario is if delta G is positive, you're just going to monotonically increase and you're never going to phase separate. And the second scenario is if delta G is negative, you're going to initially increase as gamma R squared, and then at large distances, decrease as delta G R cubed. I'm going to set pi equals 1, sorry. And here there's an activation barrier that you need to cross, called the nucleation barrier, F star, that occurs at a given critical nucleation radius, R star, which is the maximally unstable size of a droplet that you need to go through in order to create, to phase separate. So that's the classic picture in water when there's no chromatin around. Now, if you look at what these droplets are doing to chromatin, all of them seem to exclude it. So if you look at the chromatin density close to the droplet, it shows a drop, meaning that it has pushed out chromatin. There are a few exceptions to that, but I'm not going to discuss them here. So chromatin is just the DNA in its unpacked form. So it's the many chromosomes that are unpacked and that fill the nucleus.
Wasn't the droplet made of that?
Depends on the situation.
Say again?
Any DNA is called chromatin. That can be order or disorder or liquid.
Yes, I'm going to simplify things here. So here's chromatin. Here's the nucleus. There's a lot of chromatin. And the picture I'm telling you about now is that in order to nucleate a droplet, you need to push out the chromatin and exclude it. And this chromatin is a polymer gel. It's made of, like in the human genome, for instance, there's 2 meters of DNA packed into 10 microns, that is the cell nucleus, so it's pretty dense. And excluding this chromatin has a mechanical cost.
How do you look at chromatin here, just out of curiosity, I don't make no difference. You're looking at bacteria, at algae, or humans?
Eukaryotic cells, humans typically, mammalian cells.
Humans are much more organized than bacteria.
Yes, yes, of course. Bacteria don't have a nucleus. I'm focusing on this picture here, and essentially for the rest of the talk, it's going to be human cells or mouse embryonic cells, which is almost the same. Okay, so you need to, in order to create this droplet of size R, you also need to exclude the chromatin, and so there's a cost associated to that. And you can make a simple model to how much energy it costs using, so you want to use elasticity theory for that because there's a mechanical cost to deforming the chromatin. And it's a highly nonlinear elasticity theory because these droplets become much larger than the initial pore size. And so asymptotically, there's a number of different ways to define nonlinear elasticity, but they will all more or less fall into the same scaling. So let me drop all the 4 pi's here so that I don't get mixed up. The elastic cost will always scale as the elastic modulus times the volume. And the only assumption that I'm doing to get this scaling here is the fact that the stress at the boundary of the droplet while the droplet is expanding, the stress is constant. For instance, if you have a linker in the DNA that you need to unbind in order to grow, that's going to be what
You're also assuming G to be constant, right?
I'm also assuming G to be constant so far.
It's a big approximation.
This is a big approximation. But let's look at what implications this approximation has. So now the interesting thing is that the scaling of this bulk free energy term and the scaling of this volume term is the same. So essentially, you're just shifting the phase boundary. That was for, before it was for delta G negative. Now, what you're going to have is something like that. That's for delta G smaller than minus G. And this is for delta G larger than delta G larger than minus large G. You're only shifting the phase boundary. And so it becomes easier to nucleate in the regions where the elasticity, the elastic modulus, is smaller.
Delta G is negative for you here, right?
Yes.
So I'm just trying to figure out, you're not putting absolute values there, do you?
No, delta G, this term should be smaller than minus G in order to overcome the elastic barrier to nucleation. So
G is positive, and delta
G is positive, delta G is negative, yes. I mean, delta G can be positive too, but then you're never going to get phase separation.
Just trying to figure out your relation there. Okay.
So what this predicts is that the chromatin will displace the phase boundary. If you look at the phase diagram, and let me write it as a temperature or the inverse interaction strength. This is delta G equals 0, delta G equals 0. So above that, you're in the mixed phase. So this is chromatin, and you have the phase separating proteins that are all dispersed in dilute. Below that, you're nucleated. So this is only going to happen at delta G smaller than minus G. Because so this was without chromatin. This is with chromatin. There's an extra cost to deforming the chromatin. And indeed, if you look at the correlation between where the droplets appear when you autogenetically induce them, so when you turn on the light, and how dense the chromatin is, which is a good proxy for how stiff it is, then they actually appear where it's the least dense. There's a strong correlation between density of chromatin and where they appear, which is not particularly surprising. Another interesting effect is that if you have a wall in which there's a much stiffer region in which and you turn on the light, so there are stiff regions in the nucleus called heterochromatin. Let me draw this as, so there's a stiff region here, and it's soft all around. Then if you turn on the light and start nucleating droplets, they're going to nucleate everywhere where it's soft enough that they can nucleate, because here the barrier is too high and then they cannot nucleate. It's too stiff. But then there's going to be competition between all of these. They're going to grow and what is called ripen, meaning that there's going to be diffusion exchange between these. And the ones that are close to the heterochromatin are going to be favored, because there's a huge reservoir of monomers that cannot phase separate. And so my prediction is that these ones will get a competitive advantage, and you will have, in the end, all droplets localizing close to the heterochromatin and forming this kind of flower-like patterns that we observe indeed. So this is...
It's just a stiffness relation.
It's a stiffness relation. It's not just the stiffness, but it's also the variation of the stiffness in space that controls where the droplets will nucleate and grow. So that's what I could do with this extremely simple model of chromatin.
This theory, you're describing something. Certainly you've seen this in experiments. So what's the test?
So one of the tests is looking at the chromatin density, for which there are markers of chromatin density. And so looking at the nucleation rate, so how many droplets appear per second per unit volume, as a function of the chromatin density. And what we find is a very nice anti-correlation between the two, meaning that really it's much easier to nucleate where the chromatin is sparse than where it's dense. That's not directly quantitative because we don't have an access to the elastic modulus. There's a set of experiments that have been done by the group of Eric Dufresne in ETH Zurich, in which he's looked at phase separation, liquid-liquid phase separation, not in cells, which is very hard to control and to benchmark quantitatively, but in fluorinated oil in polymer gel, for which you can measure exactly all the mechanical properties. And this scenario is very well reproduced in that case.
What's an alternative explanation? Just to understand, I don't know what the different models are. This looks like just common sense. Am I missing something, or what's?
It's mostly common sense. It's putting a simple equation on a common sense explanation. So 1 second. One of the predictions here is the scaling. The fact that it does not change the nucleation barrier. It does not change only this, but it completely changes the phase boundary.
Okay, so I see this is just parameterizing what we know, but is there some alternative explanation of this
so
phenomenon?
for instance, if you thought that chromatin stiffens strongly under stress, which it might because it's a polymer gel, then you would get a different scaling here, and you would never get phase separation. So
I'm asking a different question. I'm asking, the phenomenon is known. Let's suppose the phenomenon is known, that you have this description. So what other explanation could there be of this, that you're competing with yet other explanation?
Chemical repulsion.
I'm sorry?
Chemical repulsion. There's an increase of surface tension between, so saying that this is the parameter that changes when you're in chromatin. That would be I think the data excludes this because it would change the kinetics, but not the thermodynamics. So this was what we could do with a very simple model for chromatin. Now, I've told you about this critical nucleation size, which is typically when a nucleation is possible, it's going to be order of or below tens of nanometers, so that it's not too cooperative to nucleate a new droplet. And you have to compare that with the scale at which the assumption of homogeneity of the material was made here. So actually, chromatin has a typical distance between polymers that is in 10s to 20, 30 nanometers, just by looking at how much there is in the nucleus and how long it is. And so actually, at this scale, at the scale of nucleation, chromatin is going to be extremely heterogeneous. What that means is that this model is not at all what you see when you're nucleating. And so for that, I'd like to introduce a newer model. So that part is, this is published work in collaboration with experimentalists. What I'm going to present now is really predictions that I've made on the behavior of these systems. And I'm going to complement this free energy relation here by trying to make it a bit more realistic at the level of the mesh size. So if I call psi the mesh size, so the distance between polymers, then we're still going to have this delta G r cubed plus gamma r squared plus G times the elastic cost. But now I want the elastic cost to be 0 when you're smaller than the mesh size, because then you can nucleate, you can be in the interstices of chromatin, and so there's no need to pay any elastic cost. So I'm just going to introduce a heaviside function of r minus psi. And then I want two different scalings to be reproduced. I want that at very large deformations, you get back this r cubed scaling, because that's the phenomenologically predicted behavior at large deformation. But at small deformation, I want my theory to be compatible with linear response, so with linear elasticity theory. So there's going to be essentially two terms, psi r minus psi squared, that's linear response, that's a small displacement induces a small energy cost, plus r minus psi cubed. And there are prefactors here predicted by the theory, but I'm going to drop them for this talk. Now, that's a slightly richer equation than before.
Sorry, how could they be predicted? They are just unknown.
If you assume linear elasticity theory, there's a 3 here. That's just geometry. And this part is unknown, and it depends on the strain, the yield strain of the materials. So how much you need to deform it before it starts yielding and the stress does not increase anymore.
But those are two unknown parameters. I mean,
There's a 3 and there's an alpha, which is of order 1.
3 is times the G, right? The 3 is the dependent.
Yes, yes.
G you don't know.
Yeah. G you don't know. G you can
Don't know any of them.
G you can measure experimentally or estimate experimentally by putting beads inside the chromatin and dragging them using a laser, optical tweezer, and measuring the force displacement responses. And that gives you order of magnitude of G that is of the order of hundreds to thousands of pascals. To give you a comparison, this is megapascals, hundreds of megapascals. So these are very soft materials, which is typical for biology. It's called soft matter for a reason. Now, the interesting thing about this equation, it displays a much richer phenomenological behavior than the previous one. And in particular, there's a dimensionless number. H, I will call it H, which is the elastocapillary number for this network, which is G, elastic modulus, times psi, the mesh size, divided by the surface tension. So it compares how heterogeneous your network is, how stiff it is, to the surface tension penalty. And what I predict, and that's really a new prediction, is that if you add H as horizontal axis here, then you have two distinct behaviors. If H is smaller than 1, then you get the same scenario as before. You transition from dilute phase to phase-separated phase in one step through this transition that I call nucleocavitation. Meaning that you nucleate a droplet, create a droplet out of the solute, and at the same time cavitate it, create a cavity inside the chromatin that is going to push the chromatin away. And that's a first-order phase transition. What I predict that is somewhat surprising, I think, is that if now H is larger than 1, you have this different transition where the phase boundary looks like this into a phase that looks qualitatively very different, which is many tiny droplets phase separated into the interstices and that do not grow. And I call this phase nanodroplets because they're nanometer scaled. So these ones nucleate, they phase separate, and they're small, high density regions, but they don't grow. They're going to stay stuck at the mesh size scale because of elastic repulsion between chromatin.
So just out of curiosity, I haven't looked at those numbers myself, but this sort of bulk limit, is that good enough for the nucleus? Have you put the numbers to figure out?
Which bulk limit?
Basically, all this expression you have is sort of bulk. You're not using the surface of the nucleus, basically. Is the nucleus big enough that it can live on this limit?
The nucleus is 10 microns, typically. That's the order of magnitude. The mesh size is 10 nanometers. So this phase knows nothing about the nucleus size. You really don't.
But the other one may know, right?
This one goes to microns. It's still 1,000 times smaller in volume.
I'm just curious. Maybe you're right. I'm just curious. You
It's,
have thought about this thing when you.
I think among the many uncontrolled parts of this theory, the size of the nucleus is not the least controlled part. There are many of the numbers that we have only a relatively poor estimate of. I think the assumption that the nucleus is infinite is not a terrible assumption here. So I've told you that there's this.
Sorry, just to understand, if I just take the term which is proportional to r squared, I'm just trying to understand why h equals 1 is somewhat special. I would have naively thought it might be 3 or something like that.
Yes, I should have explained that. Before, so if I plot again f of r as a function of r. Yes,
this equation. I just want to understand your symbols first. Is there a 3c? Is that what that says?
3 psi, yes.
And then there's an alpha?
Yes, I was just, let's forget about the 3 and forget about the alpha.
Oh, okay. What? That's what I was asking. Is the 1 1? Or is it order
1.
Okay, now you put an alpha there.
Yes, and now it's 1. Alpha is a phenomenological yield strain. It's typically 0.5 in artificial networks.
I just want to understand your symbols. Now you took away an alpha there. Is there an alpha there?
Alpha was a dimensionless number of order 1.
So I'm supposed to read off that h from your function f of r, right?
Okay, now I'm going to take it out of here too. I was adding too much detail for this talk. This h can be read here, and the value of h controls the sequence of shapes of the f of r function when you lower delta g. If h is smaller than 1, you're going to get the classical nucleation, the usual nucleation term, in which when delta G when delta G plus G is positive, you have a monotonically increasing curve, and when delta G plus G is negative, you have this bump, and then it decreases. If the H is larger than 1 now, you have the same initial case, and then you have another range of regime in which there's a minimum a global minimum in the free energy function at a finite size, and then at very low delta G, you get this shape. And that you're supposed to read out of this. You're just asking whether there is an intermediate minimum at some R larger than psi. Yeah, you can, I mean, this is of course analytically solvable. It's just a cubic equation. So
is anybody trying to do experiments, trying to change nucleolus to see all these regimes?
Okay, so
I'm just curious. I haven't followed it closely.
This is a prediction that I made a month ago.
No,
I'm in discussions with Cliff in particular in order to see whether we can get into this regime. Let me explain why it's not completely simple.
That's what I'm asking.
Yes. So if you look at numbers in the cell nucleus, the elastic modulus is hundreds to thousands of pascals, as I said. The mesh size is larger or approximately equal to 10 nanometers. And the surface tension is of orders of 10 to the minus 8 to 10 to the minus 6 Newton per meter. And that gives me, if all in all, and if I put the numerical factors, it gives me H of the order of 0.1 to 100. So the range of parameters that the nucleus plays with is this range. Both of the phases seem to be relevant. Now, there's a complex hard part, which is that these droplets are never going to be resolved optically. They're small puncta of the orders of 10 nanometers, 20 nanometers. And so if you're in this phase, it will look like you're homogeneous and you have not phase separated. The other thing is that increasing the interaction does not help you, because this line is vertical here, meaning that either you're going to go through this transition and you're going to phase separate, to have nanoscale phase separation, or you're going to cavitate. But going from this to here to there, you need to change either the mesh size, hard, or the elastic modulus, hard too, or the surface tension, may be possible, but we don't know how to do that.
I don't know. But if you change cations or stuff like that, maybe you can change it.
Yes, maybe indeed you can change the surface tension. So
it's cool.
I think it's a neat prediction. It's not going to be easy to confirm experimentally. Also, if you look at easier experimental systems, such as oil in water, oil in an elastic gel, which is experiments that are being done and in which this behavior was well observed. And you look at the value of H, it's now 10 to the minus 4 to 10 to the minus 3. So you're very deep into this regime, and there's no way you're going to get the level of heterogeneity that you need to get this physics. So this is really a property that is observable only on heterogeneous enough and stiff enough networks, such as chromatin, and not like chemical polymer gels, which are pretty homogeneous. And you need very low surface tension that is specific to, that is pretty distinctive of protein droplets. What is it good for? Well, there's, I've told you very briefly about the fact that there's this observation of transcription droplets that appear, and that seem to be transient, that do not coalesce when they get close to each other. And that seem to have very high dynamical, and that remain very small. And one possibility that I'd like to explore is whether these transcription droplets are consistent with this phase. So that's an idea of the type of predictions I can make just using extremely simple theories, but not much simpler than, not much worse than the data is, I would say. And yeah, I think, yeah, I'd like to say thank you for your attention.
Thank you. Thank you.
Eduardo, do you have any questions? Eduardo?
There is a problem joining the meeting from the Zoom client. Seems like we've lost. Okay, I should have said that earlier. I've seen that a while ago. Yeah, the camera is moving. The problem is with the...
Okay, well, it's too late now.
Oops.
Where is that being transmitted?
Why don't you start to... You got a question?
Yeah, we missed it while there. Just a naive question. So you said that there you will not be able to optically resolve the yellow dot, but you would still measure this because you would just see the concentration of the yellow stuff disappearing, right? You would not see the dots,
No,
but you would see...
not if the... I mean, you would see a homogeneous distribution of dots instead, but that you cannot resolve. So it would look the same as a phase separated.
It would look the same as in the top?
If you look from very far, these two without your glasses, they look the same. Because all these points, the total intensity is the same.
But can't you measure in the middle where there are no dots? No.
Well, if there's I mean, you have to imagine that this is like 100 nanometers, and the optical resolution is like this is blurred at this scale.
I see, okay.
I mean, of course, there are higher resolution microscopy techniques, which...
will be a very tough experiment.
Sorry? It will be...
You could put some probes and do confocal, but this will raise the difficulty with the experiment a lot.
So there are experiments with transcription droplets that reach resolutions of 10 to 20 nanometers that are done by Ibrahim Cissé at MIT.
Yes,
and
I just saw someone talking for him there.
Can you at least look into that?
He's trying to do. The problem is Ibrahim is doing this confocal on much simpler systems. So it's a question how you bring this stuff you can do on simpler systems to larger systems.
Okay, I have quite a bit of respect for the
No, he'll do it.
Yeah.
Ibrahim is a tough guy. I know him since he was in Illinois. So basically, and then basically he has been on doing these things. But the question is, there's a lot of technical things, and he's the right guy for that. There's no question.
Yeah. I was discussing with him last week.
Can you at least look into that phase and see that you don't have the higher radius droplet? Or?
I mean, one of the hallmarks of this phase would be that if you remove the chromatin, it phase separates. But if it's there, you don't see phase separation. And yeah, that's a way to look for it. And I think so far there's been a strong selection bias in papers published towards those in which you actually see something. It's hard to say, okay, there's a big result. We did not see anything here. I'm discussing with experimenters.
People are going to see that very soon, so it's good to have theories and predictions. So
yes.
The experiments are coming. Like you said, Ibrahim or other people, there are lots of people there. So they're coming. There's no question.
It's an incredibly hot field. Scarily, I would say. But so far, I think this is new.
Okay. All right.
Any other questions?
Thank you.
Thanks a lot. This?
Okay, not to put labels, but this is biochemistry, no?
No, it's biophysics.
Biophysics.
That's what we call it.
I'm a physicist, so it's biophysics.
But for example, the people who tried...